Good day everyone, welcome to the course on next generation sequencing technologies, data analysis and applications. In the last few classes we have looked into the analysis of variants and the different types of variants. So, in last few classes we have focused mostly on single nucleotide polymorphisms or SNPs and small indels. So, in this class what we will do, we will look into the analysis of CNVs or copy number variations and structural variants. Okay, so let us get started. So, this is the topic that we will be discussing today, methods for analyzing CNVs and SVs, and we will talk about this in very brief, okay, some of the methodologies and the idea behind these detections. Okay, so these are the keywords that will come across CNV, CNA and SV. Okay, so the structural variants they can be classified into different subclasses. So, these SVs they differ from SNPs and small indels. So, these SVs they have a part which are called CNVs or copy number variants. So, here we see only change in copy number. Okay. So, we will talk about this in bit more detail, what we mean by these CNVs. Then you have the other class which are non CNVs. So, again you have some of these different non CNVs, again we will discuss them in the next few slides. And then you have some complex SVs or complex structural variants. They can be combination of these non CNV SVs or CNVs as well. Okay. So, they are combinations of the CNVs, non CNVs, or different combinations of non CNV SVs as well. Okay. So, we call them as complex SVs. Okay. So, what are these copy number variations, or sometimes we refer to them as also copy number alterations or CNAs. Okay. So, they are actually changes in copy number of a segment of a genome. So, this segment could be a gene or could be multiple genes, right, a small region of a genome. So, there could be gain or amplification. So, this is the general term. So, we can use the term duplication. So, where we have increase by one more copy, right, instead of having one copy you have two copies now. So, that will be duplication, but the general term is gain or amplification. So, you can have increase by more, right. So, it is not just 1 to 2, it can be 1 to 3 or 1 to 4. Or you can have loss or deletion. So, maybe the gene copy or a region of a genome can be lost. Okay. So, here we see decrease in gene copy number. So, how do you identify this and how do they look like? So, for CNVs deletion, so imagine this scenario. So, here you have the reference genome sequence, here you have this gene fragment A or gene A, and then in the sample that you are working with, and you compare that with reference, and you see this gene is not present, okay, or this segment A is not present in your sample. Okay, this is deleted. So, this would be a deletion event. Okay. So, we will talk about how we identify these different SVs in a moment. Okay. What are the methods that we use. Then you have duplication. So, here as I have mentioned, duplication means you have from 1 to 2. So, here you in the reference you have a single copy which is A here, that is shown, and in the sample you have double copy, right. So, you have two copies. So, the region A has duplicated. Okay. So, this could be generalized. So, you can have instead of 1 to 2 you can have 1 to 3 or 1 to 4. Okay. So, that will be general, we generally call them as amplification. So, why are we looking at this CNV CNS? Because these are actually very important events in the genome, right. So, there are many examples of these in disease samples. Also have the non CNV SVs. Okay. These are not copy number variants, these are other type of structural variants. So, let us talk about those briefly. So, the first one is insertion. Okay. So, what happens in insertion? So, you have the reference sequence, you can imagine, and in the sample you have another fragment of genome inserted, right. So, that was not present in the original genome, it has been inserted, X region or X gene has been inserted from somewhere else. Okay. So, this will be an insertion event. You can have something called inversion. So, here you can probably see the images and probably you will understand this clearly, right. So, you have this reference with this gene A in this direction, and in your sample you see the gene A direction has reversed, right. So, we have change in the orientation, right. So, the nucleotide sequence that has changed. Okay. So, the direction has been reversed. So, that is why it is called inversion. The next one is the translocation. Okay. So, here is the reference. Now, translocation is a bit more complex. So, imagine you have these two genomic regions, one in blue, one in red. Translocation means one part of the genome has been transferred from this blue region to the red region and one part from the red region to the blue region. Okay. So, that could be the translocation event, and you can see this here in your sample. You can see these blue regions, right, they are kind of separated out now, and you have the red regions. Okay. These are simple SVs. Now, there could be many more complex SVs, and these are also found in human genomes. Okay. So, the first one is called paired duplication inversion. Okay. So, in short we call it as dup in dup, right. So, you can kind of remember what is actually happening, right. So, you will have duplication towards the end, right, which means A will be duplicated, this region A, and this region C, this will also be duplicated, right. So, you have two duplication events, so that is why paired duplication, and then you have inversion event. Okay. So, this you have created a copy of A here, you have extra copy of C here, and this region in between, this ABC region, this is inverted. Okay. So, this direction changes. So, that is what we see here, right, the direction has changed. So, if you read the nucleotide sequence, you will see the direction has changed. Okay. So, this will be your paired duplication inversion. You have duplication event as well as inversion. Okay. So, the duplication is happening at the ends, and then the whole fragment is inverted in between. So, similarly, you can have something called paired deletion inversion. Okay. So, again we have a short form, so that you can kind of remember what is actually happening or connect this. So, it is the opposite of what we discussed now, right. So, paired duplication inversion. So, here you have deletion events, right. So, at the ends, right, you see the region A is deleted and region C is deleted, whereas the intermediate region, this B, is inverted. Okay. So, two deletion events and one inversion event, right. So, what you see is this is a combination of this copy number variation, right, the earlier one as well is a combination of copy number variation with a non CNV SV, right. So, it is a combination of both. Now, you can also have something called paired deletion duplication inversion, right. So, here what you have is the reference is this ABC gene. Okay. Now, compared to the earlier cases where you have either duplication at both ends or deletion at both ends, here at one end you have deletion and at the other end you have duplication, right. So, if you see this in the reference, you have ABC, the A region is deleted, right, but the C is duplicated. Okay. So, you have created two copies of C by this event, but A region is deleted, right, you cannot find any A here. Okay. So, this is what is shown here, or you have another possibility. Okay. So, A is duplicated, okay, and C is deleted. Okay. So, here in the lower part you see this A is duplicated and C is deleted, and then of course, in between you have the inversion. Okay. So, this inversion event is there. So, that is why it is called paired deletion duplication inversion. So, you have deletion as well as duplication and inversion, right. So, you have three events occurring together. You can have other types of complex SVs. So, this is here is one is called deletion flagged inversion, right. So, del inversion or indel. So, if you look at the reference sequence, it is the A, B, right, these two regions, and what you see is that one region is deleted and the other region is inverted. Okay. So, you have one deletion event and one inversion event. Okay. So, you see deletion of gene A here or region A here and inversion of region B, and in this case you have inversion of region A and deletion of region B. Okay. So, this is another type of complex SV. So, we can look at the other one, right, which is the duplication flagged inversion, right. So, we talked about deletion flagged inversion, now we have duplication flagged inversion, and you can probably guess what it would look like. So, you have this AB reference sequence, and in the sample you have a duplication event as well as an inversion event, right. So, in this case the region A is duplicated and the AB region is inverted, and in this lower case here you have region B amplified or duplicated and this AB region inverted. Okay. So, you have two events now, duplication and inversion here. All right. So, there are different types of SVs, and you can probably find or other types of SVs as well if we look for them in the human genomes. So, how do we identify them from the genome data, right. So, this is one very important area, right. So, we have now talked about different types of SVs that could be identified in human genome. Now, the question is how do we actually identify these structural variants or SVs. Okay. So, we will talk about some methods that allow us to identify these structural variants. Okay. So, before we go there, so we should remember this is a comparative analysis. So, we are working with read data from two sets of samples, and this will be set 1 versus set 2 samples, and we want to identify whether there are structural variations in, for example, set 2 samples compared to the set 1, right. So, we need this read data from two sets of samples, okay, and this is something that is really important. Okay. So, we will talk about some of the methods that can be applied to call these structural variants. So, the first method is called the read pair distance methods, right. So, the idea is actually quite simple. So, we are working with paired end sequencing data. We have talked about single end sequencing and paired end sequencing. So, here we are working with paired end sequencing data, and what happens is that the distance between the pairs actually change because of the presence of the structural variants. Okay. So, it is not just the distance, but also sometimes in case of other complex SVs you can have also the change in orientation. So, in this case, right, if you have change in the distance, that might actually tell us okay, there might be a structural variant present in the sample. Okay. So, what we do is we compare the actual distance with the expected distance. So, the expected distance is derived from the reference. Okay. And if you have significant deviation, right, so we can identify okay, there are probably SVs that are present. Okay. So, let us take a couple of examples and see how this method will work, right. So, let us imagine, right, this is our reference sequence, right. So, we have this paired end reads, right, and this is the distance, expected distance. Okay. So, this is the reference, and then we have the sample genome, right. So, this is our sample that we are working with, and now imagine at this position we have an insertion. Okay. We get this insertion here in this position in the sample, there is an insertion. So, what will happen is that the reads that are mapping here, the distance between these pairs will increase. Okay. So, the reads that will come from here, right, this distance will increase because of this presence of this insertion around here. Okay. So, this is something that we can see, right, and then we will say okay, there is probably an insertion here. Okay. So, the expected distance is something here like this, but the actual distance will be something like this. Okay. Now, let us think about the deletion case, right. So, what will happen in case of deletion? Okay. So, imagine this is the reference genome, and and imagine, right, this region is deleted in the sample sequence. Okay. So, what will happen is that if you are mapping these reads, the distance between these reads will change, right, will be reduced because this region is deleted now. Okay. So, if we observe this kind of change, we can say, okay, there is this difference in the distance, and that might tell us, okay, there is probably an SP that is present in there. Okay. All right. So, now what will happen? So here are some tools that kind of utilize this kind of method, right. So, paired and the distance method, right. So, paired and reads. So, I have given you the names and the links, right. So, you have this SV detection tools, and of course, there are more tools out there, and you can explore them to understand how they actually work. There are some differences, but the general principle is the same. Then you have a second type of method, which is called the split read methods. Okay. So, we will talk about them in a moment. Okay. So, how does this work? Okay. So, what happens is that these are the reads that actually cover the structural variant breakpoint, right. So, breakpoint is the region where you have these structural variation events happening. Okay. And we can again look at this in case of insertion, right. We are taking very simple examples, right. So, here is the reference and here is the sample with an insertion, right. So, this is the sample and imagine this is the breakpoint, right. This is where this insertion event is happening. So, this is the breakpoint, right. This is the SP breakpoint. Okay. Now, imagine there was a kind of a read, right, that actually maps across this position. Okay. So, you can imagine you have a read here that map across this position. So, what will happen in case of this insertion, right. So, part of the read will come from here and part of the read will come from here. Okay. So, because there is an insertion, part of the read will map to this position and part of the read will map to this position, right. This is what will happen. Now, when you are generating this sample and you are mapping back to the reference data, what you will see is that you will not find any reads that will actually map across this SP breakpoint, right, because the reads from the sample sequence, when they are generated, right, you will get these continuous reads like this. So, you will not get any read that actually will map across this breakpoint, right. So, this is something that you can identify, right, and say, okay, this is expected, but we do not see this kind of reads, right. So, this kind of shows this that probably this is where the insertion is happening, and of course, then you can identify also the insertion that is happening. Okay. Now, for deletion, we can again think about this using this example of reference genome, right. Okay. So, you have reference and this region is deleted. So, this is the SP breakpoint, right. Okay. And here you will, in this sample, here you will find some reads, right, when you are sequencing this sample, sorry, this is sample, right. So, when you are getting some reads from this sample data and you try to map it against the reference, what you will see, this part of the reads will map at two different locations, right. So, one part will map to this location and the other part will map to this location. Okay. So, this kind of mapping differences will arise, and based on these differences you can then identify, okay, there is probably as SP breakpoint here, right. This has actually this will indicate that there is a structural variant that is present in the sample. Okay. So, I think one idea you probably get now in general is that we can use this distance between reads or this split mapping of reads to identify occurrence of certain SVs, right, and then of course, we can dig deeper and we can identify the actual SV that has happened, whether it is insertion, deletion, etc., depending on different cases and scenarios. And in some cases you can have very complex cases, and then probably again you have to look carefully at the data. So, here are some tools that actually use these split read methods. Again the links I have given, so you can look up and see how these methods are implementing these split read methods. Of course, there are more tools out there, and their implementations are slightly different, right, but the principle, general principle is the same. Okay. The other method, the third method, is called the read depth methods, and this method is mostly employed for looking at the copy number variations. Okay. So, we have talked about copy number variations, and so we can use this read depth method for copy number variation. So, what we do here in these methods is we look at ratio of read depths between two sets of samples, okay, and depending on that we can say, okay, there is a change in copy number. So, what you will expect, right, if you have let us say a gain or amplification or duplication, you will have more reads coming from that region, right. So, which means in turn you will get more depth in that region. Okay. So, you can take a very simple example of this. Right. So, let us say we have this region in the reference, and this region is duplicated, okay, in the sample, right. So, we have two copies of this region in the sample. Now, when you sequence this sample, right, so we will get these reads that will be mapping or coming from these regions, right, and when you do the mapping, right, all these reads together, they will map against this location here, right, in the reference genome they will all map to this location. Okay. And then if you now compare the read depth in the reference versus the sample, you will see in the sample you will have more read depth, right, because you have two regions which are generating these reads, right, that map to the same location in the reference genome, right. So, by identifying this change in read depth, right, you can then say, okay, there is an increase in copy number or a decrease in copy number, right. So, in case of deletion or decrease in copy number, you will see reduction in read depth because you will have lost some regions of this, right. So, maybe there are two copies of region in the reference and one copy is lost, then you will see reduction in depth, right. But if you have amplification or duplication, you will see an increase in read depth, right. So, based on this you can then identify, okay, there is a copy number variation around this. Of course, there is a lot of statistics as there, but this is the very simple idea behind this kind of call. Okay. So, as I have said, like higher read depth may indicate gain or duplication, and whereas if you see reduction in read depth, that might indicate loss or deletion. Okay. So, this method is mostly used for detection of CNVs, right. So, you cannot use this for other types of SVs, etc. So, we have illustrated these read depth methods, and here are some tools that are out there that you use these read depth methods. Of course, there are many more, and again I have given you the links, you can actually go and read about them, you can see the implementations if you are interested. For few more tools, right. So, for example, the BCF tools CNV, right, this can be actually very useful when you are doing the CNV calling, right, we will see. The fourth method that relies on the assembly based method, right. So, the idea is actually quite simple here, is that we do this genome assembly of reads of two sets of samples, right. We will talk about the algorithms later on, how do we actually do the assembly from read to the genome, right. And if you see any difference in assembly between the two samples, this might indicate presence of SVs, right. So, if the outcome is different for two samples, then you probably can say, okay, there is some SV present, that is why we are getting different assembly. Now, one of the limitations of this method is because of the computational requirements. So, genome assembly is a very complex process, it requires a lot of computational time and resource. So, maybe this is not the best way to go about it. Right. Here are some tools that actually implement these assembly based methods for SV detection, and again you can look up. Now, we can take these methods and you can have some sort of combined observations to actually identify different cases, different scenarios. Okay. So, we will discuss them briefly. So, we will start with deletion, right. So, let us say we are working with the short read data, right. So, we have the short read mappings, and you can see we are looking at paired end reads, we are looking at split reads, right? We have talked about these methods, and again what are the scenarios that we will see. Okay. So, in case of deletion, right, you can see like we have the reference genome now here, and then we have the sample genome. Now, what will happen is that in case of reference genome, right, R1, R2, right, if this region is deleted, the distance between them in the sample genome will be reduced, right. So, there will be reduction in the expected distance, right. And similarly, if you have reads that are coming from the sample genome, right, what will happen is that they will map across this deleted region. So, there will be split mapping. Okay. Again, this is something we have discussed already. If you take the long reads, you will have very similar observations. So, long reads might be covering this full region. Now, when you map this long read against the reference genome, you will see this split mapping. Okay. This is what we are going to see. What about the assembly based method, right? So, if you have assembly, in case of deletion, you will see this gap in assembly, right, between sample and reference. So, this region will be absent. So, we will not be able to find any corresponding region in the sample. Okay. So, we will see these differences in the assembly process. So, we can then combine these methods and perhaps we can come up with different tools. So, continuing this discussion against the duplication for duplication events, right. So, again we have the short read mapping data, it is the long read mapping data and the assembly, right. So, in case of duplication, what you will see again in the reference genome, this is duplicated. Now, if you look at this paired end data, right. So, these arrows in red color, right, their orientations will change, right. We will see this kind of orientations changing. Okay. And what it means, we will probably see discordant mapping in the results. Okay. And then again we have this reads, if we coming across this break point, right, this one in violet color, right, this will show split mapping in case of reference genome. When you are mapping the data against the reference genome, we will see this kind of split mapping. For long reads, we will see this kind of again overlap in the mapping, right, because one region has been sequenced twice, right. This was one region in the reference. So, this has been sequenced twice. So, you will see this kind of overlap in the mapping, and again if you look at the assembly part, you will see this kind of overlap in the assembly data, right. So, this will mean, okay, there is some duplication event that has happened in the sample genome. Similarly, we can extend these to, for example, inversion events, right. Again, there are different scenarios without going into a lot of details. You can actually look up and think about it, right. So, again inversion events, you will have different kind of scenarios, but again the distance, orientation of the reads, split read mapping, these things will change, right. So, similarly, if you look at the long reads, you will see this kind of change in orientation, mapping process, as well as if you look at the assembly, again the orientation, etc., this will change because of the inversion events. So, what I will do is I will encourage you to actually think about these scenarios, what will happen if you have inversion, deletion, etc., very carefully, and maybe take a piece of pen and paper and think about these situations and draw the outcomes yourself. So, that will actually help in better understanding. So, you can have insertions of extra regions, right, in the reference sequence, which is denoted by this orange region here. This region is inserted, and what will happen is that they will generate some reads from this sample, which will not be able to map against the reference genome, right, because this sequence is not present in the reference genome. And similarly, you have the same issue with long read data, right. So, again you have this read that will map the inserted region, but part of this read will not be able to map in the reference genome. And similarly, you can think about the same way in the assembly process, right. If you are comparing the sample assembly and the reference assembly, and you will see in sample that some sequences are extra and that are not present in reference. Okay. So, these kind of situations will describe, right, which type of SV is happening. And similarly, you can think of translocation, right. So, translocation means you will have change in kind of combinations, right. So, again you have some regions that were not present, and again you can think about, right, all these what will actually happen, right. If you are generating certain reads, some reads in the translocation, right. So, let us say two chromosome, part of two chromosomes have fused together, right. So, they have translocated, they have fused together, and you are generating these reads R1 and R2, right, from the sample, and when you are mapping them back to the reference genome, they will map to 2 different chromosomes, right. So, this means we will get this discordant mapping, right. Those results then become important for this kind of scenario. You can think about the split read case, etc. What will happen in case of long reads? Again, part of them, part of the read will map to one chromosome, the other part will map to another chromosome, right. And again, this discordant mapping will help us identify this SV, and also will help us understand the type of SV, right, that has happened. So, what we will talk about is that there are an array of methods that actually take this combined approach, right. They will actually combine all these different methods that we have discussed, right, the read depth, assembly, etc. They can combine some of these methods and generate a much more accurate result. So, they will combine maybe 2 or 3 of these, right. So, paired and read plus split read and read depth. Again, assembly is not really favored because of the computational resource requirements, etc. And it can, and so these methods can take any 2 of these or all 3 of these and can help generate a very accurate picture of the SVs. Okay. So, when we combine these methods, we get the benefits of all these approaches, right, and we also get lower false positive calls. Okay. Here are some tools that actually implement this. They kind of take this combined approach and they get much more accurate results. So, here are the references that we have used for this class, and in this class, to summarize, we have seen different types of SVs we have discussed, and these which are observed in genomes, right. These are not just made up or thought of, but we actually identify these different types of SVs in the genomes, and we have discussed about different methods and tools that can help us identify these structural variants. And what we have also seen, that combined approach brings together the strengths of each of these methods, right. So, we get a much more accurate view of the structural variants that are out there, and we get best results and we reduce the number of false positive calls. Thank you.